Hello, welcome back to my science channel, I guess now, because the only videos on my channel are of science videos. Today, I'll be talking to you about DNA replication. DNA is what makes us us. There's over billions and billions and billions of codes in our bodies to make us who we are, what we look like, and what's inside of our body. It makes DNA is what controls our whole entire body and what we look like. To start off, we should talk about the structure of DNA before we actually talk about the actual DNA replication. DNA is what we call a double helix form. It's this spiral, you imagine it as a spiral staircase going up. It's spiraled around each other and it has base pairs. And the leading strand goes from 3' to 5', while the lagging strand goes from 5' to 3'. With in between these strands, there's these hydrogen bonds of pairs of AT and CG, or thymine, adenine, cytosine, and guanine. The hydrogen bonds are very weak in between the two. That's why I just drew a little broken squiggly thing. They're very weak for the purpose of being broken down and to be replicated. Now that we know what it looks like and the order of the pairs and the order of the strands, let us move on to the actual replication process. Before we move on, I can't stress this enough, let me tell you that T always pairs with A, C always pairs with G. Never any other pairing. If it does pair with something else, then it is incorrect and it's proofread before it's actually finished replicating, so it's always proofread and always T and A, C and G. Okay, now that we know about the pairs and the shape of DNA, we can talk about how it's actually replicated. To start off, we need to talk about some enzymes. These are the major enzymes that are used in DNA replication. There's plenty more, but these are the major ones that you should know about. We have helicase, which is the unzipper of the DNA. It goes through and breaks all the hydrogen bonds. Then we have primers. Primer is what starts off the base pairing for the DNA polymerase, because the DNA doesn't know, or the DNA polymerase doesn't know where to go. And that's why we call this the starter. And the DNA polymerase, like I said, it's what builds the rest of the pairs onto the DNA strand. And that's why it's called the builder. And then you got ligase, which is what they call the gluer, because it glues together fragments of DNA's strands which I'll talk about later on in the lagging strand. But these are the four major enzymes that you should know about. And now we can move on to what it looks like to break down a DNA strand and which enzymes go where and what it looks like. Okay, now we are going to start talking about DNA replication. I'm going to talk about each step and what it looks like and then put it together all at the end. The first step is the unzipping of the DNA. Like here, this is what the actual DNA strand in its double helix form looks like with its hydrogen bonds and everything. But the helicase comes in and breaks those weak hydrogen bonds, unzipping it, causing them to be separated. Enough room for another strand of DNA to be made to come through. But like again, I said that each base pair has to be the same, the A, T, C, G, and that plays a critical role. And when the DNA polymerase comes in and attaches the things to it, it would have to attach a T to the A, a G to the C, and so on and so forth. But the helicase is the first step of unzipping the DNA, allowing room for the new DNA replication process to begin. Alright, the next step is the starting of the DNA replication. After the helicase unzips the DNA strand, we have the primase, or also known as the RNA primase, comes in and starts a position of base pair for the DNA polymase to find where it needs to start. Also, this is known as the origin. In the leading strand, which is this top strand that I have over here, it starts at the beginning. The primase starts at the beginning in the leading strand. If I was talking about the lagging strand, it would start somewhere else in like the middle, for example, because it's harder for them to go from 5' to 3' than it is for them to go to 3' to 5'. So to make it easier on themselves, they go from 3 to 5 no matter what. So that's why sometimes they'll start in the middle for the lagging strand. But anyway, we're going to talk about the leading strand and how it starts at the beginning, at the origin. And the primase starts the first couple of base pairs. I only drew one just to save some space. But it starts the first base pair of T pairing with the A, and that's why we call it the starter. Alright, the next step to DNA replication is the DNA polymase coming in and adding on the pairs. So the helicase breaks and unzips the base pairs. The primase starts for the DNA polymase to find a starting point, or the origin. DNA polymase comes in here and just starts running through, attaching pairs, attaching pairs, attaching pairs. And that goes all the way through however long the DNA strand is. And also, the DNA polymase is also the proofreader of his own work. You know how, like, if you're writing an essay, you're going to go back over and make sure that it looks good. The DNA polymase does the same thing. It goes back and makes sure they didn't mess up on the base pairs. But it just goes through, keeps on trucking through, adding base pairs to what is already on that strand. Earlier I brought up the enzyme ligase. The reason we aren't talking about it in the leading strand DNA replication is because it's not really used. The ligase is to glue together fragments of DNA. Like I said, the lagging strand, the primase starts, the primer or primase starts somewhere in the middle, not really at the beginning like how the leading strand does. So on the lagging strand, the primase starts like in the middle, say for right here, and then the DNA polymase would come through and fill in just a section at a time. Those sections need to be put together and connected, so that's what the ligase is for. It comes in and connects those sections on the lagging strand. But we're not talking about that because we're talking about the leading strand, but that is what the ligase is for. Alright, and now to put it all together in one. The helicase comes in, unzips the DNA strand, breaking the weak hydrogen bonds. The primase starts at the origin, gives the DNA polymase a starting point to start. They start off the first couple of base pairs, and then the DNA polymase comes through and runs through, adding base pairs, and adding base pairs, until the end of the DNA strand. The DNA polymase is also what proofreads its own work. It goes back over and makes sure that it added the right base pairs, and this goes out through the whole entire DNA strand. Next, I'll show you what it looks like all put together after DNA replication happens. Alright, and now for the end of DNA replication. You should have two identical DNA strands. They're identical because it was the same base pairs. That's not hard to mess up. But DNA replication is one of the most complex things that our cell does, and this all is happening in our body at all times, and we don't even know about it. But DNA replication is used to have identical DNA in different cells when it splits up in mitosis or when it comes together in meiosis. but you need to have the identical DNA because you don't want a random change out of nowhere. It's got to be the exact same. That's why they are identical. But again, DNA replication is like the most complex thing that our body does, and this is all happening in what we call the interphase of cell growth, or the cell cycle, the life of a cell. This DNA replication is always happening in interphase, or also the S phase, but that is for a different video about cell cycles. But this is the end of DNA replication. I hope you learned a thing or two. To recap on everything, you need to know that DNA is a double helix form with the base pairs of AT and CG, and they always go together. Can't stress that enough. And to break down a DNA strand, you need helicase to unzip it, and then to start the replication, you need a primase or an RNA primase to start the replication process, and then you have the DNA polymerase coming through and adding all the rest of the pairs, and then you have ligase to bring together the DNA fragments, and once it all comes together, you should have 2 identical DNA strands. And this is the end of my recap. So this is the end of my video, and this is only my second video on my YouTube channel, so give it a like, and it's the only reason that I have a YouTube channel now. But thank you for watching, and I hope you have a great day. Bye.